The revision video for genetics part 2, you would need to really watch the genetics part 1 video to able to apply this. And remember, particularly for genetics, this is only a limited amount of information. You need to be able to fully revise this and go over content in your own time. Variation is a sum of differences between individuals or different. Variation is caused by two factors: genetic factors, which are inherited in your genotype, or environmental factors, for example, skin color, which can be darkened by the sun, or weight, which can be affected by diet, exercise, and many other factors. We obviously rely on the production of new alleles to cause variation, and this is caused by mutations that lead to a random change in the basis of DNA. Most of the time this does not give an advantage, but most mutations have, mostly they'll have no effect, and sometimes they can be negative, but they can also be positive. Mutations can lead to new alleles. Meiosis can lead to a new combination of alleles due to crossing over and random assortment, and new combinations of alleles come from random fertilization between gametes. Discontinuous variation then is one of the two types you need to know. So discontinuous variation is when you belong, your variation category is one of a category, sorry. So blood group is the best example of this, where you're blood group A, blood group B, blood group AB, or blood group O. But other things could be classed as discontinuous, such as tongue rolling. Continuous variation then falls into particular groups, and as a result we represent it with a bar chart, and there should be a gap between each of the bars there. Normally it's caused by a singular gene, in case it's monogenic, and not normally affected by the environment. Variation then is where there is a variation in the population with no distinct groups. So height and weight are a good example. These are controlled by many genes, so we call them polygenic, and each gene has an additive effect. This is also heavily influenced by the environment. Continuous variation. Continuous has no distinct categories, whereas discontinuous does. Continuous tends to be quantitative, so you do a line graph, whereas discontinuous tends to be qualitative. You're controlled by a lot of genes in continuous, but discontinuous is by very few genes, and it is strongly influenced by the environment, whereas discontinuous is unaffected by the environment. To represent continuous variation, we use a normal distribution curve. So you can see here most people, or most examples, will be in the middle because there is a more common, a favourable level. But as you go down, let's say this was height, less and less people are smaller, and less people are towards the extremes. And that again is because most people, there's a favourable optimum characteristic level which most will acquaint to, and there'll be less people at the extreme. To investigate variation, one thing that we need to be able to do is look at the population, but it isn't possible to measure the whole population. So what we would do is a sample, but this may not be representative. We may get the results we want by look, but we have to do this by taking multiple readings and taking a mean. So realistically, if we wanted to know the height of people in the UK, we would have to probably try and get the height of at least, say, 10% of people. There could be, however, sampling bias, where the experimenter chooses measurements, and we avoid this by random sampling. So when we measure investigate variation, that we would obviously calculate a mean, and you should know that you do that by adding the sum of all the individual levels up and divided by the number of repeats you have. Calculate the standard deviation, which is a measure of spread of data around the mean. So here you can see the bits of data. So if we have 40, 45 and 50, the mean is 45. There's a wide standard deviation, so it might look like this graph here. But if this one 44, 45, 46, the mean is also 45, but there is a much narrower standard deviation. So standard deviation is variation around the mean. There's also more variation, and therefore it will be less precise if you have a wider standard deviation, so you'd have less confidence about your result. Different examples of variation. Skin color is continuous variation because it goes for across a range of colors and levels of pigmentation. You can use skin color to look at how different variations may be beneficial or less beneficial in different areas. So we think skin color, life we think evolved in sort of Africa, and we think dark skin was natural in those parts of the world because there's more melanin. Melanin has a higher UV protection but means you absorb less vitamin D. Pale skin, though, would become more advantageous for people as they moved away from the equator because there is less UV protection but can absorb lots of vitamin D even in low sunlight. So then let's go back to the genetic crosses. So the first one is dihybrid crosses. Last time, and dihybrid cross is where we look at two copies of two different genes and their inheritance. So during the production of gametes, two copies of the hereditary factors segregate, and you get one factor from each parent. But we're now looking at what happens when we look at more than one gene. So dihybrid crosses, more than one gene has been looked at, so we're looking at two genes at a time, and those genes are not linked on the same chromosome. It was Gregor Mendel that worked out this information. We take this for this genetics for granted, but he did it without any knowledge of genes. So if we have a look here at an example, we have peas that are wrinkled and green. So the green is recessive. So it's got a small g, and wrinkled is recessive, so it's got a small r. And we've got two sets, don't forget, because it's a purebred one. So little r is the fact that they're wrinkled, little g is the fact that they're green. For yellow and round is dominant, so capital R, capital G is for the fact that it is yellow, which is the dominant allele. Through meiosis, they'll make the gametes, so obviously they'll pass on either of each letter, so that would be both, would have all the gametes would have a little r and little g for that one, big R, big G for that one. So the first filial generation would be heterozygote, heterozygotic, or I can't get the word out, sorry, but it would be it would have different alleles. So you would have yellow because round, because R is the dominant one, and that's round, and yellow because the capital G is dominant, which is yellow. What happened then is we would go on and create a second filial generation. So you would need to be able to draw that for this example being bred. Look at that. We know the first generation would be that. Second generation, this is what you would expect as a result. So you breed them together, but again, you could now have a combination of alleles. So you could have capital R big G, capital R little g, capital little r big G, little r little g, and the same for the other one. You would then place both of these into the gametes, because don't forget they're inheriting each gene, and they are one allele of each gene. And what you should always find when you do a dihybrid cross, which has no linkage, etc., is that you'll get a 9:3 to 3:1 ratio. So 9 double dominant, round and yellow, 3 where there's one dominant and one recessive, and 3 again, and then 1 where there's 2 recessive characteristics being shown. And you can see this in Mendel's work. He didn't get exact numbers, but he got these sort of ratios when he crossbred the F1 generation to generate the F2 generation. And again, you might get different examples, so feel free to pause the video here and see what you would get, but remember that outcome that you would expect. So Mendel's second law, as a result, was that he was able to explain his ratio because he said that one gene does not affect the other, and this is summarized in his second law, which states that alleles of different genes are inherited independently. This, however, doesn't follow when we do autosomal linkage. So this is genes that are linked, but in the 22 pairs of chromosomes that are not the sex chromosomes. 23 chromosomes contain your genes, or pairs of chromosomes, Thus, the chromosomes contain many genes. Genes on the same chromosome are called linked, and therefore there is an increased chance if you're looking at two genes on the same chromosome, they'll be inherited together. And if there's no crossing over through chiasmata, they will separate together, and they don't do independent assortment in meiosis, so don't follow the law of segregation. Genes on the same chromosome are called linked. Here we have some examples of this. So if you look at this chromosome here, okay, in the past we've talked about the random assortment of independent assortment. So here you've got the big A, little a, big B, little b, little b. So you're always going to inherit one brown and one blue, so you're going to get all the different combinations possible. But if we're looking at autosomal linkage, the A and B are on the same chromosome. So what that means is even when these chromosomes are pulled apart into sister chromatids later on, that A is still always going to be inherited with that B. That A is going to be that B, that little a with that little b, that little a with that little b. And the only way that could change is if crossing over occurs and the B is swapped with another capital B, for example. So in this case, law of segregation is. So an example of this is in Drosophila melanogaster. And you can see here that because there is linkage, the capital G is always inherited into the gamete with the capital N, and the little n is always inherited with the little g. So the recessive ones are inherited together, the dominant ones are inherited together. Now, as a result, that will affect the genetics. So you will always have capitals together and you'll always have recessive. So 2 out of every 4 will be dominant, 1 will be dominant but heterozygous dominant, sorry, 2 of them will be heterozygous dominant, and 1 of them will be recessive. So you're still getting a 3-to-1, but there's a difference in the way that it's inherited. And as I suppose, as a result, you're not getting the 9-to-3 to 3-to-1 ratio of a dihybrid cross you would expect. So the last part is chi-squared. So this is a statistical test to identify whether the difference in results that you are seeing from the expected ratio is significant. So when we're looking at more than one gene, which are not linked on the same chromosome, we can carry out, which is what we call a dihybrid. Who said came up with these numbers, he came up with this law of segregation. By finding these findings. So what we've said is we should have a 9-to-3 to 3-to-1 ratio. But in real life, are these numbers actually following that ratio? And that's what chi-squared is to do. So chi-squared is saying, is 315 the number we should be getting? And if it isn't, let's say you should have 318, is that difference significant or not? If it is significant, then we will not accept these findings, and if it is significant, isn't significant, we can say that difference may just be due to chance alone. Mendel's, with large samples, it's unlikely he ever got this pure 9-to-3 to 3-to-1 ratio. So a statistical test is to see, is that difference from this ratio significant or is it not significant? So if it's not significant and it is due to chance factors alone, this is a null hypothesis. So we're saying the difference is not significant. And that's what we want in this case, which is different from many times we look at a null hypothesis. If the significance is significant, so it is actually significantly different, and there is a reason for this, we then accept the experimental hypothesis, and we actually don't want that in this case. To do chi-squared, data must be in categories. So, for example, our 4 categories of 4 potential phenotypes. It has to have a strong biological theory behind it to test for. It has to have a large sample size. The data should only be in raw counts, so you can't do it with percentages, for example. And there is no 0 scores when. So if we carried out this experiment, we would look at this and we'd go, right, let's find out the actual numbers. And we would want to have the null hypothesis. So we want to be able to say there is no statistical significant difference between the observed and the expected data. Any difference is due to chance. And if that could be supported, we would then be accepting the work of Gregor Mendel. Have a look at a worked example. So here we have some numbers. We've got 169 yellow and brown, dominant, so that's one that should be 9. 51 there, so 3, 3, and 1, which is 14. So if we do this, the first thing is we need to work out the expected ratio. So we've obviously got a 9 to 3 to 3 to 1 ratio there. So that equals 16 if I add all those up. Okay, and therefore if I take 288, which is the total, adding all of these up together, I should get 1 would equal 18. So I then do 18 times 9 to work out the ratio for this one, 18 times 3, 18 times 3, and 18 times 1 for that. What gives me is a set of data. So my observed ratio is the numbers I actually got, okay, and then 18 times 9 is 162, 18 times 3, 18 times 3, 18 times 1. So then I do observed minus expected, which is for the chi-squared equation. So observed minus expected, 169 take away 162, 54 take away 54, 51 take away 54, and 14 take away 18, which will give me that data there. I then square this, which removes the negatives. So that gives me 49, 0, 9, and 16. And then I divide this by the expected number as well. So I'm going to do 49 divided by 162, which equals 0.3, etc., etc. The sum of all those together equals my chi-squared number. So again, you need to be able to work your way through that. Once I've got chi-squared, I then compare it to the critical value is here. So my number is this one. So if chi-squared is greater than this, I reject the null hypothesis, which is what I don't want to do in this case, because that tells me there is a difference that is significant between Mendel's values and the observed. And if it is lower than the critical value, I accept the null hypothesis. Now what I do here is I look for a critical value at 5%, which means we're 95% certain our data is correct. And if my number is lower than these critical values, I can accept this data. Now I had 4 different groups, my 4 different categories, which means I have 3 degrees of freedom, which means my data needs to be lower than that number there, which it is. And that means I can accept my null hypothesis that any differences between the observed data and the predicted data or the expected data is only due to chance alone.